Good morning. My name is Gabor Betlendi. I'm the co-founder and chief commercial officer of Parabase Genomics. I want to welcome everybody to this informational exome sequencing webinar for patients and families. I have first confirmed that everyone can hear me. Oh, thank you. I have a confirmation everyone can hear me. So first of all, to thank the Global Genes Projects and SWAN for making this possible. I think without their strong support and efforts for fundraising, it wouldn't be possible. So I greatly appreciate them involving us in this project. So today we're going to, next slide, please. So today, in this webinar, we're going to spend the first half an hour going through what exome sequencing is, what Parabase Genomics is as a company, basically walking through the testing and consent process and make sure that the families are comfortable with how reports will be reported, what are some of the deliverables they will be getting, and of course, what are the limitations of testing. Because I think in this day of age of Google, we feel that some of the expectations can be misaligned, especially when it comes to exome sequencing. So we make sure that everyone's comfortable with that. Now, this webinar, I think the guests on the call will be muted, but there'll be probably about 15, 20 minutes, half an hour at the end where you'll have an opportunity to ask questions. And I would encourage you to submit questions through the chat option on the webinar or jot them down, and we'll open the mics at the end and handle them as appropriate. Okay, next slide. So my name is Gabor Betlendi. I'm the co-founder and chief commercial officer at Parabase Genomics. My background actually is in biochemistry. I do have a master's in social work. It's a long life story, but it has brought me to this point where I have started my own company along with Dr. Bhattacharjee, who was one of the pioneers in exome sequencing. He was recognized for his work in probably Science and Nature in 2010, I think, as one of the breakthroughs of the year. So I've been in the academia industry for a long time. And interestingly enough, probably for the families on this call, my 9-year-old son, actually does have a rare genetic condition. Just by serendipity. I won't be with those details, so you have a lot of camaraderie when it comes from my perspective. And really hope we can at least give you more information through this testing process. Joining me today is Heather McLaughlin from Harvard. Heather, would you like to introduce yourself?
Sure. So I'm Heather McLaughlin. I'm from Harvard, and I also work at the Laboratory for Molecular Medicine. So I have been doing a lot of exome and genome sequencing work here at the laboratory, and hopefully if anybody has any questions about the testing process or about what to expect, I can help to answer some of those questions today.
Thank you, Heather. And just for the participants, maybe Heather was a little bit demure, but the Laboratory for Molecular Medicine, along with their director, Dr. Heidi Rem, is one of the leaders and pioneers in the U.S., perhaps even in the world in exome sequencing, so we're very pleased to be working with them as well. Let's go to the next slide. So Parabase Genomics is somewhat of a startup company. We were founded in 2009. We're based here in Boston. We're NIH and privately funded. We did mostly exome and targeted sequencing for neonates in the NICU and pediatric type cases. We normally don't handle all the types of cases because these cases, as Heather will attest to later, are quite demanding. As I mentioned already, both Andy and I have come from companies, Agilent Technologies and Nimblegen, that have developed exome sequencing from a technological perspective. And I think we're somewhat blessed to be using the technology now to actually help real families such as yourself. We developed some novel methods for collecting samples from dry blood spots, which we won't be using here. And And if you're in the Boston area, you're welcome to come visit us. We'd be happy to give you a tour. And we partner with several hospitals. That probably, if there are folks online who are local, Brigham is probably the biggest one we partner with right now, although there are other ones that we work with. Okay, next slide. So I think it's important to understand basically the 3 steps involved in exome sequencing, and to distinguish a couple of things. First, we are not sequencing the whole genome, okay? And I'll go through what exome sequencing in greater detail, but it essentially involves these 3 boxes of blood sample collection where a doctor has requested the test, okay? And that's a very key distinction because this is not a direct-to-consumer test. It has to be ordered by a physician, okay? Next step is sequencing, and I'll go through some of the steps of that. And finally, it's basically data analysis and reporting, which is pretty much what the physician and I think the families on this call are really interested in. We will go a little bit into these details just so we have a general sense of it, so it gives you enough ammo to ask us the relevant question and to other providers you might be working with. Okay. Next. So what is an exome? So like I said, it's not the genome. Actually, it's a very small part of the genome. Really only about 1% of the genome. And if you look at the top, and if you look at this bar and this yellow and gray zebra-looking thing, you'll see that this is a chunk of DNA. Now here it's a cartoon stick figure, but it's trying to show you that it's a double-stranded piece of DNA, okay? And when these chunks of DNA, there are parts that are called introns and parts that are called exons. Exons are the regions that we're most interested in because those are what we call the functional parts of the genome. That is, that's where the DNA is translated into proteins and where most of the action is. And matter of fact, if you you were to look it up, about 85% of the known disease-causing mutations are in the exons. And while there are mutations and what I would call academically interesting things happening in the introns, a very unexplored area at this point. And so most of the scientific community has been focusing on exome sequencing for the last 5 or 6 years because it's very fruitful in terms of finding novel mutations, disease-causing mutations, and whatnot. But it's interesting that there are about 30,000 in the human genomes. And in this particular test that we're providing, we're sequencing all the 220,000 exons that make up those 30,000 genes. So in other words, there are only about 8 to 10 exons, depending on how large the gene is, sometimes more if it's a large gene, representing each gene. Okay, so we're basically enriching the exome to take out the exons, the yellow portion that is shown here in this apple slice, and we're only sequencing the things that are relevant to us. Okay? To the next slide, and I'll show you more of what I really mean. Oh, there's the slide after that. There's another slide in step 2. So these 3 steps include sample collection and isolation. So next slide. There are basically 3 types of sample collection, sample types we can collect. There's dry blood spots, which we do collect right now. There's whole blood and saliva. For this particular project that we're doing with SWAN and GGP, we're only collecting whole blood, but we do have abilities to perform the test of dry blood spots and saliva. If you've already been to your physician, there's various methods used to collect the blood. I show here in the middle 2 photos of a collection of whole blood from an adult. They will do a venous puncture to collect the blood. And at the bottom in the center section, you see that there's basically a wrist being done on a newborn. Now, depending on how old the baby is, what the veins look like, etc., this blood collection extraction sites will be utilized. Right, you essentially see sort of the result. Usually it's a smaller tube than this that the blood is in for a baby. This tube is actually more for an adult. And from that, we use various DNA extraction methods to arrive at a tube that's at the bottom. You see this cloudy thing at the bottom? That's actually genomic DNA purified from the blood sample above. And that's pretty much what your whole genome comes down to, is that little blob in the right corner in the clear tube. Okay. Next slide. That process only takes an hour or two at most. Okay, next. Now, I don't want you to get overwhelmed with the method of biology here. The intention is not here to go too deep, but I did want you to get a sense of what's happening with the genomic DNA that we do purify from these blood samples. The last is blue globule is pretty much the genomic DNA that I showed in the prior slide, that blob in the test tube. And I mentioned before, genomic DNA is represented by exons, the thousands of exons. And we basically, various adapters allow us to purify only the exons from these genomes, capture them on these next-gen sequencing machines, sequence only the regions of interest. And I'm very happy to provide more literature on describing this entire process or talk one-on-one with the families. But just so you have the general understanding that we're not sequencing the genome, basically enriching just the coding parts of the genome in a very high parallel fashion. I'm putting on this instrument which is called Illumina HiSeq instrument. Okay. Once the DNA goes through extraction, sequencing, we end up with basically raw sequencing reads. Okay, and these are basically, if you will look at them on your computer, be basically rows and rows of A, T, C, and Gs. Okay, just on and on and on and on and on, millions of bases. Okay. These strands of DNA have to be aligned to a reference genome. In other words, an already sequenced genome, so we can compare to that. So we can see the mutations that are different between the reference and the known sample or the unknown sample that you have given us. And that comparison allows us to call the variants or mutations relative to the reference genome, basically generate a report of mutations or variants, allows us to generate a medical report. A lot of other steps that go within each one of these red, green, and purple arrows. And again, I'm happy to provide as much information as the families need. But this is just a high-level view, again, of the steps that we go through to generate the medical report. Next. One question that Heather has brought to my attention, and I had actually forgotten because I've been in this business too long, is that once these mutations or variants are generated by softwares, there are ways of classifying these mutations into different categories. And it's very important to follow guidelines to do this. And actually, Heidi's laboratory that Heather's from at Harvard is one of the pioneers in this field. Published a paper in 2007, along with other groups, of course, with the American College of Medical Genetics, that's ACMG, and there's standard guidelines that most laboratories should be following if they're doing exome sequencing, and we do follow these guidelines. My intent here is not for everyone to become experts in the variant classification algorithm, because I think that's why we have Heather and Harvard on board. But if you go to the next slide, the takeaway is that we're following the ACMG guidelines, which is very important. And they pretty much, the mutations that the report is going to generate is going to fall into one of these four general categories, whether it be deleterious, that is causing damage to an exon or a protein, and possibly resulting in a disease phenotype, or it could be benign, that is, nothing will happen. And there's various ways that there's other subroutines that Heather and other labs use to determine whether there's enough evidence to determine whether they're deleterious or benign. And another group which, if they're not deleterious or benign, they can either be predicted to be deleterious or predicted to be benign based on softwares and algorithms we use. Okay. Next slide. Now, not a real case that I'm showing you, but once we classify those mutations into appropriate buckets, we generate a medical report. Your report could look something like this, although they will be different depending on the type of mutations that are called, if anything is called at all. On the left, you have an example report from Parabase, anonymized the information, but it shows an abnormal finding for Escobar syndrome, which is a rare genetic mutation that results in basically webbing of the limbs and hands. In this particular case, this baby was probably about 8 weeks old. Fortunately, the baby was diagnosed early, but she had pretty much the fatal version of this mutation, was discharged subsequently. But the point here is that a report will tell you which one of these buckets the mutation fall in. These reports will be sent directly to the physician who ordered the test. We'll go through those details in a little bit. And inside, I'm not sure if we need to spend too much time on this, but it's a sequence from Brigham and Women's Hospital. Again, this is the Escobar syndrome case on the left. The point that I'm making here is that times diagnosis can be made where you have a positive diagnosis, or it could be a situation where you actually don't have a diagnosis, and there's no treatment for the diagnosis. In this case, the good thing was that the baby was discharged to go home and didn't spend weeks in the hospital. Next slide. Now, I think, so next few slides, I'm going to go through the consent form. And I know we have already received one sample from one other family, so thank you. On the right, you see an image of the consent form, which I'm sure GGP has or will share with you. It's all downloadable. Well, actually, this one's not downloadable from the website because it has the GGP logo in the right upper corner, and that's how we're distinguishing these samples. But the point of the consent form is to make sure that we have the approval of a physician to test the sample. In other words, it comes from a physician and not from a family directly. But as importantly, I think it provides you, the family, with the details about the test and the associated risks. And in the next few slides, I'm going to go through each one of the sections on the consent form where you will have to put your initial to consent in or out of those sections. And while we may not be able to go through, we'll find enough time to go through it, so I encourage you to ask questions. But I bring this up because oftentimes when we are confronted with these forms, as I do when I buy a house or a car, this is a 9-page consent form. So it could be a little overwhelming reading everything without us giving you the cues here as to what the significance of each consent step is. Okay? So it really gives you the choice of what you want to learn about the results. Okay, next slide. Okay, first, let's go through some of the limitations. Exome is not the be-all and end-all. So it can't technically possible to capture and sequence 100% of the human exome. And there are many reasons for that, again, I'm happy to go very deep, and I'm sure Heather is, as to why, but some of the high-level reasons is that some portions of the genes are very difficult to sequence using this method, even traditional methods. And the reasons like genes called pseudogenes that are very similar to each other. They're very similar. They're very difficult to distinguish when we go into the analysis portion of classifying the mutations. So for this reason, we can only expect to cover about 90 to 95% in an exome. So that 1% of the genome that we're sequencing, we can only technically efficiently sequence 90 to 95% of that 1% that I showed you in the prior slide. Okay? I think there are animation after this one. Yes, thank you. Now, so what it really means is that on the right-hand side, the exome test that we're using is called RareDX. Will provide to you this 120x average coverage. What this means is that for every mutation or variant that we're calling, will be sequenced 120 times. So very high confidence that the mutation that we're calling is analytically correct. Okay? But it does not necessarily mean that we're going to have 100% diagnostic yield. So that's why here below that we put, for a given exome, if you look at the literature, you can expect a diagnostic yield of about 30 to 40%, depending on the case. Okay, that's sort of the general consensus right now in the scientific community, and it's been published in at least 2 or 3 papers that I can share with you if you're interested. Okay, so again, if we were to take 100 families or 100 cases, about 30 or 40% would have a positive or some kind of diagnosis that could act on or manage. Okay. For the Global Genes Project, normally our turnaround time is 6 to 10 weeks. But I think for GGP and the SWAN project, we have accelerated that. And so we expect to have results back in 4 to 6 weeks. Now, there's a caveat that we discussed with Global Gene Projects, is that the way the samples are coming in, for example, we received 1 sample last week already. We need to group these samples in groups of 4. So there may be a stretch in this number of weeks based on when the 4th sample comes in, because we're grouping them 4 at a time. Okay. So while each individual sample could be done in 6 weeks, or until we get 4, we're going to hold them back until we get 4 at a time. Okay. The other thing you should notice on the left is that exomes cannot detect chromosomal type aberrations right now. We can detect deletions, and I think Heather can speak to this later. We also can detect copy number variations. So copy number variations are single base pair mutations like I showed you earlier, but they're larger changes in the DNA, hundreds of bases or more type changes, so very gross level changes. There are other tools that can be used to detect those, like chromosomal microarrays, but be aware that this exome will not detect chromosomal triallelic inheritance and all the other things listed here. And I think we can go through what each one of these means, but again, this is listed in fine print on the consent, and you should be aware of it and ask appropriate questions as you sign off on it. Okay. Slide. There's also a little checkbox on the consent form of secondary findings. These are findings that are not related to the reason for testing. You know, you came, let's say you came for testing reason A because you suspected a particular group of disorders that your child has, but they could have a different disorder. Okay, so you should be aware that you may or may not want to be informed about carrier status of recessive disorders. So these are mutations that are sort of that is, your child carries these mutations, and the right combinations, should they have children later, could result in a manifestation of those phenotypes. Okay, you may choose not to want to know about predisposition to late onset disease. That is diseases that come later, like Alzheimer's, Parkinson's disease, breast cancer, and there's a well-defined group of those we can discuss with you. You may want to know whether you have a predisposition to cancer. Okay. And these are all things that you can opt in or out of on the consent form. We can provide them in a separate report, but we would encourage that those requests come directly from the referring physician, and we can go through them and see what's appropriate given the situation. Now, lastly, the third thing is that the way we structured this exome and actually the exomes, we're not reporting out variants associated with the drug metabolisms, okay? Kind of variants associated with modest changes in the risk of common diseases, and those like cardiovascular, diabetes, and other more common disorders, or variants of unknown significance. That is, if we find a base pair change in your exome, which is not enough well characterized, or there's not enough evidence in literature to make sense of it, we're not going to report that because there's really not much the physician, the family, or anyone else can do about it at this point. Okay. So there are risks with this test. And one of them is that you may learn about the non-paternity status of a family member. And again, you have a choice as to opt in or opt out about knowing these things. Okay. You can also choose to dispose of or retain the samples for research purposes. Okay, so you sent us the blood sample, and we have more than enough DNA left after we perform our testing that we could use it for research purposes later on. You have a right to tell us to dispose of it or allow us to use it for research purposes in a way where we would anonymize the sample. That is, no one would know that it came from your particular child or family. And then, of course, cancel the test prior to the test setup. Test setup is essentially to the DNA extraction step. Okay. So let's say the child was drawn, you sent the sample, and you had a change of heart. You have that window of a few days until you start the DNA extraction process to opt out of it. Okay. Next slide. And that's really the high level that I wanted to cover. It's very challenging to do these webinars because I'm not getting any feedback. So I'm going to open it up to any questions that have come up and allow you all to ask us questions and allow Heather as well to chime in as appropriate.
I think just remind everybody, if you do have questions, you can just type them in the chat box and just make sure that you at least select all panelists so that we make sure that either Laura or I can see and answer your question.
Yeah, at this time there are no questions, which worries me or makes me happy. But I guess so. We can have a few minutes and see if some pop up. Are you seeing any other questions coming in? Katie, can you advance the next slide, please? Now, you have additional question from myself or Heather. I think we were kind of bad in not giving your email address, but if you contact me you can drop our Parabase a note at parabasegenomics.com, and I'll make sure that I or someone else gets back to you. So there's a question that's come up, Heather. Can I read it? Does it have to get permission to find out later developing results.
So I guess the question is, does the physician have to? So yes, I mean, it would actually, it's not really the physician's choice, it's the patient's choice to get those results, and that does need to happen during the consenting process in order for those results to be returned to the patient.
Right. There's another question. 30 to 40% figure. Two questions now. We're in question overload. Is 30 to 40% figure the percentage of all samples analyzed which have a deleterious mutations?
Yeah, I think the 30 to 40% positive figure is the percentage of all samples that are analyzed that were where the report was deemed to be positive, and so that would mean that yes, in that report they found a deleterious mutation directly correlated with the disease that the patient was presenting with. Basically, we found a mutation that would explain the disease in the individual that was being sequenced about 30 to 40% of the time.
Yes. Okay, question. Just to make sure I understand correctly, if I want a detailed copy of the testing and results, the ordering physician needs to order them. It depends on how detailed, right? So they'll get the report, which will basically a case summary at the top. It lists the disease-causing mutations and point them to literature that supports that. You're referring about the raw sequencing information. Yes, that would have to come from the physician. Now, on the consent form, as you fill it out with your physician, there is a section where the physician and you can fill out where the report can be sent to other parties. So you have the option of receiving it that way as well, but have to be involved. Okay, something on a single gene.
That was a question?
Yeah, just reading them through. Sorry, Heather. They're kind of cryptic because
okay.
It's like a chat box. So sorry, but yeah, I think you want or should I?
So I think I understand the question correct. Like certainly we can see any variation that comes from a single gene. But if the question is that you're very interested in knowing exactly what was found in a single gene, I think that's something that would need to be followed up with Gabor and his team to see exactly what was found, because as he was explaining, only certain things are going to be reported. So if there was maybe a pathogenic variant found in that gene, that would certainly be returned on the test. However, if there are other variants, maybe variants of uncertain significance that we're not quite sure about, those may not end up on the report. But I think if a physician or a patient was interested in anything that was found in a specific gene, that can be kind of something that can be followed up with afterwards.
Yeah, normally the way things work, and Heather can chime in, is that the patient shows up with a list of ICD-9 codes or potential diagnoses, and the gene that you're particularly interested in is in there. Normally, we'll look at those first. So the presenting phenotype of the patient is very relevant to how we start sort of prioritizing the genes or genes of the exome to look in for a possible culprit. But yes, we can actually take single genes or groups of genes and look at them specifically. So the next question is, so can we receive a copy of the report directly, detailed report as well? The answer is yes. Any more questions come in? Send myself a note at info@parabasegenomics.com. It's all Parabase distribution. And if for some reason you don't have Katie's or you want to get through to Katie or Heather, I'm happy to forward them as appropriate. There's one more question. Okay, another question. I would like the raw data to be able to share with other research programs to better chances of a diagnosis. Is this possible? Yes, it is possible, yes. We have to work out a way of sending you the data. We would most likely send you a hard drive or a drive, actually, because it's one file. We would send a thumb drive to the appropriate location. Continuing on more questions. Are more coming in? I'm Bob Barker on The Price is Right. I'm waiting on these things to come in, so I appreciate the questions. These are good questions. They're relevant. You know, the funny thing is probably about 5 years ago, we couldn't have answered quite a few of these, so we've learned a lot in 5 years. I think there's no more questions. One final parting comment. This webinar is recorded, and I think it'll be made available, I think, for future playback. I think the Global Genes folks will likely be in touch with participants if they would like to have them. And I want to thank everybody for participating. I really appreciate the chance of working with the families. It's truly a humbling experience, and look forward to talking to you directly. Thank you very much.
Thanks, everyone.